This is an experiment to show osmosis in a non-living membrane. It should be on around page 41 of your book. To do this, we're going to need some really simple bits of kit. We need a 20% sugar solution, or sucrose. We need a beaker of water, and we need to have a semi-permeable membrane, which is Visking tubing. Visking tubing starts out looking like this. We cut a section off and we soak it in water. That makes it lovely and pliable. We would make a knot in one end. We play about with this end until we can open that tubing up, and we will fill that tubing with the sugar solution eventually. Now hopefully what you'll notice is that it's starting to look like a sock that's been filled up with sand or something like that. But when you've got enough in it, we will be taking one bit of capillary tubing, that's this thing here, placing it in the centre of that sock and tying it on either with a rubber band or a bit of cotton. Okay, this is a really bad example for you to watch, but fortunately I prepared one earlier. So your final product will look similar to this. It's a bag with 20% sugar solution inside it and with liquid coming up a capillary tube. Okay, so previously you've seen examples of diffusion where all the molecules are free to move and they spread out and you get an even concentration. With this Visking tubing, the larger molecules aren't free to move. So what is likely to happen here is that the sugar molecules are going to stay in the bag, water molecules are free to move. Okay, I've put the sugar solution in the Visking tubing in the bag, I've started a stop clock, and I've also taken a note of the level of water that there is in the Visking tubing, and that starts off at 0. And what we're attempting to do is every 2 minutes we're going to check for a change in the level of water that travels up the Visking tubing. The water is able to move and it will try and make the concentration on the inside of that bag the same as on the outside of that bag. And in doing so, it will fill the bag up with water and create a pressure that will drive water up that capillary tube. Well, we've got the first 3 sets of results, and what you should be seeing is at time 0, the height that the water in the capillary is labelled as 0. At time 2 minutes, we're here, and the height of the liquid in the capillary has moved up, and I've made it move up 5 centimetres because I'm an idiot, but in the book it says 5 millimetres. Who'd believe that? And after 4 minutes, we're looking at 12 millimetres of height from 0. Okay, the actual results you will find in your book.